Good evening, one and all present here. I hope you all enjoyed your tea break and had a chance to network and recharge. We'll now start with our next session on cellular systematic and lifestyle mediators. I would also like to inform that a parallel session is also running, Frameworks of Healthy Aging, in main auditorium. Now I'd like to invite the session chair, Dr. Shantanu Shukla, to the stage. He is a longevity investigator and assistant professor at Department of Developmental Biology and Genetics at Indian Institute of Science. Over to you, sir.
Thank you. A warm welcome to everybody for this session. Apologies for the slight delay. We'll try and still keep all the time as much as possible so that we don't intrude too much into lunch. So we'll dive right away into the talks. Our first speaker is Professor Kasturi Mitra. She's an associate professor at Ashoka University, and she's going to talk about mitochondria and aging. Over to you.
Thanks very much for the invite. Yes, I'll try to stay close. I decided to do it a little differently. I'm a fundamental biologist in a session where we have translational interest as well. So I'm a fundamental biologist who has deep interest in translational medicine because we want to not pick up questions from the literature, but apply our own understanding to see if they fit in the patient population and also design tools to be able to address our fundamental questions in the real world. So I named the session as the talk as role of mitochondria in aging, which is a very popular topic. We've read both translation and fundamental scientists would have read a whole lot of literature on it. Mitochondria are the homeostatic cellular organelles, and there are new lenses being added to the role of mitochondria almost every 5 years. So we'll go over some of them, and we belong in that area. And this is basically, the pointer's not working, so maybe I'll use this. Yeah. So this is basically the hallmarks of aging. We majority of us may know from Guido Kumar's review, and we belong here. We study mitochondria, we study stem cells, we study senescence, and we study nutrient utilization by mitochondria. So anything, we do not study, disclaimer, we do not study aging directly, but I'm trying to convince you today that whatever we study is very much applicable to aging, and we've also initiated some projects in those directions because it's so relevant. So just to remind all of you that mitochondria are homeostatic organelles. They do a whole lot of homeostasis for the cells and their functionality, which is hardcore, bioenergetics initially for bioenergetics and ATP generation, redox homeostasis, metabolism, lipid modification, calcium homeostasis. This list is getting bigger and bigger, and then the cellular processes they impact are also getting bigger and bigger, including immunity, which is the recent hot area where role of mitochondria is being tested. Old ideas about mitochondrial role in aging all stems from Herman's idea of free radical theory of aging, which has been dejected, which has been modified, which has been tested several times over. And we have come to this newer idea of mitohormesis. So Herman suggested that if there is mitochondrial damage, that's going to increase reactive oxygen species generation, and that is going to cause aging. And people have tried taking antioxidants that actually propelled many subjects towards aging. So what was going wrong? So various researchers came up with this idea that is it that there is a certain level of stress that is good for, because mitochondria are homeostatic organelles, there's a certain level of stress that is good for the organism. The organism is well tuned to adapt to that stress, and then it does better. So that's when an organism undergoes mitohormesis, because of certain low level of mitochondrial stress, it does better to future hardcore stresses. So mitochondrial stress response basically is mitohormesis. And this applies for various age-related diseases, neurodegenerative diseases, immunity, cancer, and metabolic diseases. I'll show you projects in various areas there. So this is what we do. As I said, that we do integrated basic and translational research, and then we do microscopy, flow cytometry, genomics to be able to design our assays that would be translationally relevant. The new tool, a new lens that we study in the lab is basically mitochondrial heterogeneity. No mitochondria in two cells or even within a cell are exactly same. Even all of us are sitting here, we actually represent a huge heterogeneity as a population of mitochondria. Our mitochondria are different in different tissues for each of us. Even within single cells, mitochondria are different, and they are impacted by various homeostatic processes and various cellular milieu. So we study that. We study the heterogeneity of mitochondrial structure and function. Here you see the movies playing. Thank you very much. So here you see on the top, a single cell where mitochondria are forming network, and they are not all same. So that's mitochondrial heterogeneity within a single cell. Here you see mitochondria structure and function, network structure and redox function heterogeneity, as you see that this isolated mitochondria is trying to connect to the larger network of the cell, and the dots that you see coming and going, they are basically redox active spots. So they're getting oxidized or reduced, okay? That's what we study in a very quantitative manner so that we are able to beat the heterogeneity or understand the heterogeneity quantitatively and apply it at the patient level or a subject level to be able to get to personalized medicine. And the way we study mitochondrial structure function heterogeneity is by studying this phenomena, which is mitochondrial fission and fusion events. If you see here, mitochondria are constantly undergoing fission and fusion events. The proliferating cells do that more often than differentiated cells. And these are the molecular processes. There are certain fusion molecules, there are certain fission molecules which aid in these change of structures, okay? So we study those processes directly and their impact on mitochondrial function, and then on cellular function, and then organismal function. So here, from the literature, just to impress upon you that whatever we study, so here is basically the fission molecule, which causes a mitochondrial structure, a hyperfused mitochondrial structure. Well, all the mitochondria in one cell are fused together. If you highly activate this fission molecule, it'll convert to the structure, okay? And function will change at the same time. And it has been found that in C. elegans, in worm model, it has been found that if you introduce shRNA or RNAi strategy for this particular fission molecule, in the context of insulin mutants, the DAF2 mutants, which is very well known for having a long lifespan in worms, Cynthia Kenyon's work, if you now introduce shRNA of this particular molecule in the DAF2 mutants, it goes further. It goes further. So it goes further. And these are two different works. This is not yet published. This was published in 2011. So this is very exciting, okay? You do it in the context of the very well-known DAF2 mutants in the insulin signaling pathway. This pushes the lifespan further. That's what we are interested in from various age-related disorders, but also directly into aging. So again, this molecule can be playing a role in these two features. As you see here, these are directly from human samples. This is iPSCs generated from old and young patient samples. And this is basically centenarians, where you see mitochondria have different structure than the old and young cells, right? So possibly this is also playing out in real humans. So what we do is basically to study the basic mechanism for all these neurodegenerative, for all these age-related disorders, and we want to develop assays to be able to identify biomarkers for pushing it towards personalized medicine. And the stars that you see here will be repeated along the talk, and wherever you see stars, those are the assays we would like to try out in patients with collaborations, of course. So that's why it's very important for us to be able to showcase our work in this forum. So we wrote a review in the Experimental Gerontology Journal proposing that what we discovered, fundamentally in the lab, a particular state of stem cell which are primed by mitochondria to be important for aging. So these are adult stem cells which have 10-fold higher self-renewal ability. They have lower mitochondrial fission, so mitochondrial fission molecule has to be less active, and they have higher mitochondrial potential, which supports bioenergetics and various other mitochondrial function. And we used a flow cytometry assay, star shown here. That means this can be applied on patient samples, and we are trying this in collaboration with for some cancer patients for studying breast cancer. And these are the prime populations which can be isolated by using mitochondrial potential along with the stem cell marker. And you can see that if you do stem cell assay, they are they are tenfold higher in self-renewal ability. We have tested this in xenografts, we have tested this in ovarian cancer patients, and we were able to isolate them using this strategy. So what else we do? We want to use to develop biomarkers. We want to be able to tease out mitochondrial structure function heterogeneity in single cells. So we have developed microscopy assays where you can express structure function probes. Here it's basically a redox probe that you see here. A ratio of these two will show you the redox function. And you can do quantitative microscopy to be able to get all these parameters from single cells. Okay? And using that assay, mitosin square assay, again, star, suggesting that this can be used for a translational purpose, we are able to isolate different states of mitochondria, stem cells, which match very well with single-cell RNA-seq published in another paper. And we are able to tease out structure further, and these are details which are published already, you may want to look into. So we're talking about mitochondrial priming of stem cells by lowering of mitochondrial fission. What we realize is that you cannot lower mitochondrial fission to close to zero. So here what we are doing in Drosophila, we use Drosophila model in the lab for genetic studies as well. So here we are introducing different hypomorphs. That means they are lower active alleles of mitochondrial fission molecule, DRP1. And these are DRP1 and 2, these are mutant 1 and 2. And this KG mutant is basically the almost functional null of DRP1, right? So here you see the GSC proliferation, the germ-line stem cell proliferation in Drosophila ovary, which are clonally marked here. We are basically following the clonal cells. Here you see that they are higher, right? And this higher ability of GSC proliferation happens only in a light and dark cycle. So if you keep flies in a 12-hour light and day cycle, that means circadian rhythm you're maintaining, only there you are able to see this priming. But if you maintain a continuous light or a continuous dark cycle, that means you are basically relying not on the light, but endogenous cycle, these don't work. And this probably is playing out in here, where you see in young, when we do the egg-laying assay, because this is Drosophila ovary, we do egg-laying assay and see that in young adults, in Drosophila, you're able to stimulate egg-laying with diet. And these egg-laying are basically counted in light and dark, as I was telling you, light and dark cycle. But if you take the old flies and do the same assay, you will see that not only the diet cannot stimulate, they don't have any difference between light and dark. Right, so their Drosophila egg-laying is well known to have a circadian rhythm, and we believe that what we are going to test is whether this priming event controlled by mitochondria can be responsible for such differences. That was Drosophila ovary. In human keratinocyte line, we actually showed that fine-tuned repression of mitochondrial fission primes neoplastic stem cells. When I say fine-tuned, that means you cannot abrogate the activity completely. If you abrogate the activity completely, it's going to inhibit the stemness. But if you regulate the level of the activity of mitochondrial fission to a zone, a Goldilocks zone, it actually supports transformation processes. It supports neoplastic transformation by sustaining this particular makeup of stem cells. Okay? This is keratinocytes, and you can form tumors with these cells that are more stem-like, and the tumors of the same carcinogen exposed for the higher dose, which do not have this line, or rather this state enriched, they do not form. They form a very well-defined squamous carcinoma, whereas the primary tumors, which are very much stem-like, they are not able to form these tumors, but they are able to metastasize very well. So we were able to characterize mitochondria, again by our quantitative assays, to be able to get to the exact mitochondria structure which will support this particular state. Here you see low dose of carcinogen, high dose of carcinogen, low dose of carcinogen enriches the structure. And if you have very fine-tuned repression of DRP1, you enrich that structure. Okay? So this homeostatic zone has to be maintained for enabling very efficient transformation of the stem cells. And this is single cell RNA-seq, which actually depicts very similar comparison between these two states at the level of stem cells, which are depicted by their genomic expression. So we realized recently in the lab that this process where you're transforming the cells to generate the mitochondrial prime stem cells by this low dose may be happening through senescence induction. What a student in the lab has done is taken cells, did not let it go all the way to transform, but taken cells out of this process in between the transformation process after 7 days and done senescence assay. You see the same dose, which actually enriches mitochondrial prime stem cells, also causes higher senescence. Okay? And the dose that is not able to cause enrichment of mitochondrial prime stem cells, that causes, that can get to this higher level of senescence only after you add mitotempo. So mitotempo is a reducing agent, right? Here again, we are going away from Hermann, saying that certain level of redox is important for establishment of stemness. So if you reduce this by mitotempo, you're going to enhance the level of stemness, okay? So this intermediate level of stemness, intermediate level of redox supports the mitochondria prime stem cell enrichment. We are able to develop an assay at the level of single mitochondria now, single mitochondria within single cells, where we can get to this metric called cell relative length of mitochondrial network within single cells to be able to parse out different kinds of network, different sizes of network based on the threshold of this particular metric. Again, it can be applied to translational studies. And these are the small mitochondrial networks. These are the large mitochondrial networks, the hyperfused mitochondrial networks. So they can be interconverted. And one way to interconvert them is actually to activate neoplastic stem cells. So we've done the same assay on neoplastic stem cells, added media which induces stemness and proliferation. There we see that there's maximum conversion of small mitochondrial, hyperfused mitochondrial networks to small mitochondrial networks here. And here these are various different properties, structural properties that one can study. We realized that stem cell enriching carcinogen exposure promptly converts the hyperfused mitochondria, which are oxidized, to actually redox tune them. Here you see this particular stem cell enriching dose enriches mitochondria in this intermediate level of redox. And if you look at the structure function property of mitochondria, you'll be able to define which network characteristic of mitochondria supports that redox function, or vice versa. We have been able to use AI/ML-based prediction from various structural parameters for predicting the oxidation level. And we found that the cell relative length of mitochondrial network, which I was telling you is important for distinguishing different sizes of mitochondrial network, is the highest predictor. Okay? And again, this can be used for translational studies. This I will skip for the lack of time. And here I will show you that what we are proposing about mitochondrial homeostasis and mitochondrial hormesis, rather, to be able to establish a higher stemness gene expression by maintaining mitochondria at mitochondrial redox state at this very intermediate zone, to be able to sustain SOX2 positive stem cell activity, which we are actually testing in collaboration with Dr. Munia Shami, CNRS investigator at the University of Côte d'Azur, whether this plays out in the neurogenesis process in Alzheimer's. And Alzheimer's neurogenesis process is defective. So if mitochondria priming through this method, or rather through this fundamental biology, will be important for Alzheimer's study. This I'll skip for the sake of time, and I'll leave you with this idea that whatever fundamental mechanism we study, in the lab of various at various physiologically relevant situations for aging and age-related disorders can be tested with all the methods that we developed: flow cytometry, microscopy, and genomics tools, and can be tested on young, old, and centenarians with with and without age-related disorders, depending on what their clinical presentation is. And we can use PBMCs, which we are trying for cancer patients, and the relevant tissues to be able to test them. So I'll end here with acknowledging all of you for your patience, and my group here and the funding source. Thank you very much.